Good evening, everyone. To begin, I'd like to acknowledge the traditional custodians of country throughout Australia and recognise their continuing connection to land, waters and culture. I pay my respect to elders past and present, and I extend that respect to all Aboriginal and Torres Strait Islander people that are joining us today. So we're thrilled to welcome two leading voices in the field of psychedelics and fungi for this special Q&A on wood lovers paralysis, a rare and intriguing phenomenon associated with certain psilocybin mushrooms. Dr. Simon Beck is a medical doctor, harm reduction educator, and drug policy reform advocate. He's the Secretary of Australian Psychedelic Society. He has had an interest in mushroom identification for over a decade. He is interested in the clinical use of psychedelics and is also passionate about decriminalisation. Kane Barlow is a fungi educator and mycologist based in Melbourne, Australia. He gives regular talks on mycology, fungi conservation, and teaches gourmet mushroom cultivation. He works closely with the Australian organisation Entheogenesis Australis and is co-founder of the US-based organisation, the Entheome Foundation. He's written for Entheogenesis Australis, Double Blind, Third Wave, Microdose, and Healing Maps. Kane is the author of Wakeful Mushroom Cultivation Journal, published in 2024. He is an admin and moderator on many Facebook fungi groups. Together, they have been interested in woodlover paralysis for over 6 years, leading to a survey study in 2020. That study led to the publication of the first systematic description of woodlovers paralysis in scientific literature, and the first systematic exploration of some potential environmental and individual factors in the syndrome, written in collaboration with researchers Dr. Monica Barrett and Dr. Liam Engel. If you have any questions during the session, drop them in the YouTube chat below and we'll do our best to cover as many as possible. So good evening to everyone. So I'd first like to extend this to Kane. So what's led to your interest in woodlover paralysis?
I was fairly active at the time on many Facebook groups and from about 2015. And I guess from 2015 through to about 2020, I noticed a marked increase of questions around this syndrome, woodlover paralysis, to which there was and having a bit of a Google, it would turn up in Shroomery and a few other places, but there was very, very little known about it. And I guess it was a matter of kind of trying to cobble together a little bit of an understanding of what this was and how to then be able to answer questions in that context of being an admin and moderator to the community.
Yeah, oh, sorry, go ahead.
Oh no, and hence then Simon and I kind of started talking. But I can let Simon kind of give his perspective on that.
Yeah. First up, thanks heaps, Pip, for organising or for agreeing to sort of moderate this for us. Really appreciate it, and all the awesome promotional work you do for the Australian Psychedelic Society. My interest in woodlover's paralysis came from sort of seeing it in the wild quite a few times, and coming from sort of a medical perspective, it just absolutely fascinated me. Like Kane, the best source of information, and I'm so happy that I lived in an era where you could just Google this stuff, because otherwise it would have been absolutely baffling. Stumbling across some sort of forum posts about it, which at least provided some comfort that it wasn't something completely unique when I did see it the first time. But there was nothing sort of formally described, which just struck me as really unusual, because there were a few reports, there was a bit of chatter about it online. It seemed like a really, really interesting phenomenon, and one that I thought could pretty reasonably be foreseen to pose a pretty significant risk for a range of reasons. So from sort of a harm reduction perspective, it seemed like something that really warranted a bit of further exploration, and also just out of sheer curiosity, because it's such a bizarre thing.
Thank you very much. And I think the community really appreciates you charting that uncharted terrain in this. So would you like to tell me a little bit about what your research process has been like?
Yeah, it was a really good collaboration. Finding some really interested people. Kane has been active in the psychedelic scene in Australia for probably near on as long as I've been alive. So he's been around for a while, and had, lots of really helpful connections. I was also moderating on the PMANZ Facebook group for like mushroom ID and harm reduction stuff on Facebook. So we had sort of plenty of connections to reach out and try to track down some information. Kane and I spent a long time sitting down and sort of designing a survey from the ground up that was, in hindsight, longer and clunkier than it needed to be, but you live and you learn. And we did leave out a few important questions that, would be good to answer in future. We did that because when I first got interested and started reaching out to like clinical toxicologists and societies representing clinical toxicologists, the responses were really odd and a little bit, disheartening. One clinical toxicologist who worked for one of the poisons hotlines, which, you as a doctor in a hospital would ring if somebody came in with a poisoning syndrome that you needed advice around, was like, doesn't exist because I haven't heard of it. I thought, well, that's a really weird way to approach a question about something that we're saying, we have seen and clearly does exist, that just hasn't been described. And some of the others were saying, look, even though it sounds interesting, researching it's just going to be too hard because it involves, illegal substances, and that's both sort of academically unpalatable, but also technically challenging because you need all these permits and all this sort of stuff. So it just went into the too hard basket. Hence sort of landing on a survey design before looking at chemical analysis or anything like that, in the hopes that if we could get a description of it published, people with the sort of facilities and the technical know-how to do more of the chemical analysis and the further work would actually be interested, because there would be some evidence that it existed. So it's like, we know it exists. It's existed for a lot longer than we've been writing about it for, but now it exists because it's published in a medical journal. So yeah, hopefully people will actually look into it now, which is nice.
Fantastic. Absolutely. I think it's very much needed. So for the uninitiated, would you be able to talk a little bit about what exactly is wood-lover paralysis and how it shows up?
Yeah, sure. I don't sort of want to talk too much about the basic description of any of this stuff. I just encourage people to sort of read the paper for that sort of basic info. But to give a bit of a basic rundown to anyone who's not familiar, it is a sort of cluster of symptoms, mainly around muscle weakness, of varying degrees, affecting different parts of the body and often sort of shifting throughout the duration of the experience, that occurs after eating a range of different species known as lignicolous Psilocybe species, so the ones that grow on decaying wood matter, as opposed to the ones that might grow on grass or dung primarily. That includes Psilocybe subaruginosa in Australia, but also Azarescens, cyanescens, and a range of other related species, and has been reported from some less closely related species, or at least one much less closely related species in Japan, which is really interesting too. Typically, symptoms occur in about 50% of cases in the first 2 hours after ingestion, but in the remaining 50%, they occur sort of between then and a full day later for the first onset of the weakness, and tends to last sort of anywhere from a couple of hours to residual symptoms lasting for 1 day, 2 days, 3 days. Residual symptoms persisting into the next day was not uncommon. 2 or 3 days was far less common, and usually with less severity. Important to note that it can feel like it's gotten better and then come back. And there are certain things that seem like they make the weakness worse or better. The main thing that seems like it makes the weakness worse consistently is use of the muscle group. So generally people would describe that if they used a particular muscle group, the weakness would get worse. If they rested it for a while, they'd regain some strength. But if they returned to using that muscle group again, they'd sort of get worse again. Which again is really, really interesting to think about from the perspective of how this might be occurring, like what the mechanism might be. But that's the basic rundown. That can be associated with some changes in sensation at times as well, but primarily it's the weakness issues, and that can be really profound. It can actually have a really significant impact on functioning.
Thank you. And Kane, is there anything you'd like to add to that?
Oh, I think Simon summed it up pretty accurately.
Yeah.
Yeah.
Fantastic. So the question has been asked, is it dangerous or is it just uncomfortable?
Kane, do you want to field that one?
Yeah, it is potentially dangerous. Given some of the reports that we have from to chatting to various people, it sounds like in extreme cases it can cause severe breathing problems. So people can uh, then end up in a situation where they need CPR and being kept alive. So yeah,
Yeah.
definitely very potentially dangerous. And also with the fact that then so many people report that the following day, they don't experience it till the following day. There's then the potential for accidents in certain situations as well. One of our respondents mentioned that they went swimming the following day, and then while they were swimming, the paralysis kicked in, and they were lucky enough to swim back to shore.
Yeah. Heaps of near misses in cars and bikes and that sort of stuff, walking across roads as well.
And is there a way that you feel that you'd be able to distinguish, I guess, discomfort from a medical emergency in a situation where it might show up?
I'm just looking at the chat. I just want to mention actually that it's really interesting timing, and just want to say thanks to the person in the chat who I won't name for sharing the experience that their partner required sort of revival because they stopped breathing with Wood-Lover's paralysis recently. Not the first case we've heard. Glad she's okay. Certainly there've been at least 2 deaths that I have heard about, including one fairly recent one in Australia, and one previously in New Zealand that I think are a direct result of the weakness. It makes sense that at some point it can start to affect the muscles of breathing. If it affects all the other skeletal muscles, at some point it's going to start affecting the diaphragm. So I think that is actually worth noting. I would discourage anyone from trying to distinguish this from a medical emergency. Obviously, if you get a sudden onset of weakness of any form and of any extent, one of the big concerns is, have you had a stroke? That's the big thing to be particularly worried about now. Just because you've also taken mushrooms, it doesn't mean that you can't have a stroke. Mushrooms do increase blood pressure, albeit somewhat slightly, but particularly predisposed individuals, that increase in blood pressure, plus or minus maybe some distress from the experience itself, increasing your blood pressure a little bit further, could potentially put you at risk of having a stroke at that point in time. So my thought would be that, I know it's a difficult proposition and it's an easy thing to say without actually being there in the moment, but if you have a sudden onset of weakness for some reason, you should probably go and get to a hospital, post haste to be looked over. The reason I say that is that strokes don't always present with like that typical sort of unilateral or like one-sided weakness affecting one side of the body and the other side of the face, and this sort of thing. Is that's a big stroke in a particular area of the brain, yes, but that's not the only way that something like that can happen. Likewise, some types of seizure activity can cause some pretty odd changes in your physical experience and your sort of movement. So some of those things, when you're not sort of trained to pick them up, and even if you are trained to pick them up, if you're not in a sort of clear state of mind, it's probably not a great idea to rely on your general training and capacity. I'd say err on the side of caution. And again, that Wood-Lover paralysis itself can seemingly, in severe cases, really cause some pretty severe outcomes, potentially including stopping your breathing and causing death in and of itself. It's probably better to be somewhere that can be monitored if it looks like it's getting severe. But certainly, if someone looks like they are struggling to breathe, if someone is sort of changing their level of consciousness, becoming drowsy, particularly if they're becoming drowsy and either breathing very, very loudly or very shallow and very slow and softly, if they're becoming difficult to rouse by speech. Once you have to start shaking people, you are in trouble. There's a scale of responsiveness called AVPU, and once you're in the poo, you're in the poo. So you're either alert, you're responding to voice, that's the A and the V, or you're only responding to pain, or you're unresponsive. You know, once you're down at that other end there, really you need to be being medically monitored. But you also don't want to wait until you're there to be calling an ambulance. So I say just err on the side of caution. Don't try and make your own diagnoses and stuff like that. Wood-lover paralysis does tend to come on in a sort of non-hemispheric distribution. Like it's unlikely that your whole one side of a body is going to be weak without any effect on the other side. More often than not, it's a little bit less sort of generalized and specific than that. But the distribution varies heaps, and it changes throughout the duration of the experience. So whether it be that you're worried about a stroke, or whether it be that the possibility that wood-lover's paralysis is going to get really bad and put you at risk on its own, ideally, you know, you're off to hospital. Certainly, if someone looks like they're struggling to breathe, you're starting to do basic life support, CPR if you need to, call an ambulance without any hesitation. If someone looks like they've gotten better, even if it was really, you know, if someone has sort of looked like they've had this quite badly, and then it looks like they've gotten better, they should still be going to the hospital, because it can come back. In the case in New Zealand, it did come back, and I think that possibly led to a death. You know, a recent case that we'll hopefully write up soon here, the person had CPR for quite a long time, but looked well by the time the ambulance got there. The ambulance nearly didn't take them to hospital, but then they stopped breathing again in the ambulance. So luckily the ambulance had taken them. They then needed to be ventilated on a machine overnight to survive. So don't be tempted to just write it out if it looks like they've gotten better. If it was pretty bad, they should probably still go to hospital. Should still go to hospital.
Thank you. I think that's very, very important information for harm reduction. We've had a question come through, someone asking, could it be psychosomatic?
This does not seem to be psychosomatic. From all the responses we've had, people clearly report struggling with being able to use hands, being able to walk, and in some cases eat. So, you know, this is not typically a psychosomatic kind of experience. This is, yeah, people are trying to do things, and are unable to use their muscles.
Yeah, and the description of the symptoms is fairly consistent. Like there's a wide variety within it, but the consistency of that variation is pretty stable between subjects, like person to person, which is less typical of a psychosomatic thing. I've seen it. Doesn't look in any way psychosomatic. It looks very much like a physical weakness. The awareness of what one wants to do with the muscle or the hand or the leg or the whatever is clear. The planning of the action is clear. It is just the strength that lets the person down, and they're unable to sort of complete that action. It's not associated necessarily with a subjective degree of distress, you know, and usually psychosomatic reactions come about when somebody's more distressed or overwhelmed. You know, in a lot of these cases, people only start getting distressed after the weakness hits because they don't know what's going on. And in a lot of cases, oddly enough, during, you know, in our survey, people actually said they weren't that distressed at any point, although that did vary widely, but some people were far less distressed than others. So I don't think it's psychosomatic at all. Plus, 2 to 3 days later, you know, like the mushrooms are well and truly worn off then. And there's people who don't report ever having similar experiences without the mushrooms being involved. It's just would be a really unusual way for a conversion sort of picture, psychosomatic sort of thing to present.
Great, thank you very much. So we've got a question that's come through in the chat. So someone said, in my teens, which was in the late 70s, I took mushrooms for years and never heard of this condition. Is it something that has been presenting more recently, or have there been cases before this?
I think just the general awareness of it has kind of means that people are now talking about it more, and perhaps a lot more aware of what the symptoms are, what the syndrome presents like. I think there's a general feeling that up until recently, people have kind of written it off as perhaps oh, you were just, you were just too high, or the experience was just too strong, and it was just a matter of that you couldn't move your body. And I think now with the internet, with people being able to talk about it, there's just more awareness of it, and so people are able to better identify what's going on, and hence talk about it.
Great. Thank you. We've had another question come through via social media that someone said that it only happens to them when it's cold. Is that a common phenomenon?
Yeah, yeah, super interesting, and may give us some indication of what the mechanism is. But that was actually one of the more common things people said seemed to make it worse, which is why part of the harm reduction advice is to try and stay warm and also to get warm if you are experiencing this. Um, it, it, between the nerves and the muscles, obviously, like generating a movement involves contraction of the muscles, right? The nerve has to release an electrical impulse that releases sort of chemicals that cross over a cleft, a bit of a gap between the nerve cell and the muscle cell. Those chemicals activate receptors on the muscle cell, and that's what generates a contraction in the muscles. That gap between the two is called a neuromuscular junction. Neuromuscular junction blockers are used medically to induce paralysis for things like operations. And in operations, you try to keep people warm if you don't want to prolong the neuromuscular junction blocking effect of a lot of these agents, because being cold does actually seem to impair the recovery of the neuromuscular junction or impair its function. So it sort of is interesting that if that is the way it works, which kind of is my sort of leading thought at the moment, that being cold making it worse might actually sort of support that concept of its mechanism.
Thank you. There's a number of people who have shared their experiences in the chat. This is obviously an issue that is sort of quite, widespread, even though it's quite rare. So it's, very important that we're having this educational evening tonight. So just I've got some questions, more environmental questions now. So is there any indication that a particular patch can cause it? And is it common for it to occur in the same patch multiple times, or from, and yeah.
There's no indication that a patch can cause wood lober paralysis. It's by virtue of the fact that it's well, we feel it's highly likely that it's a compound within the mushroom. There's no physiological presentation within the mushroom itself, or any indication otherwise from growth patterns that patch is going to cause it. We have identified through the survey with questions that people have experienced wood lober paralysis from the same patch over multiple years. So by virtue of probably being caused by a genetic factor, and particularly in the case of the lignicola psilocybe aeruginosa, and related species, they survive over multiple years, unlike cubensis, which will occur within one patch, but then occur in a different patch in different years. The substrate is consistent, so that genetic individual within the substrate is consistent over time. With respect to whether it's common, we still consider it a rare syndrome. It happens rarely, but it's common enough within the overall population that it does turn up in patches in that sense. So from Tasmania through to Queensland, South Australia, and then patches overseas. So it's part of the genetic component of the overall species, but it's just unclear when it presents. So this is a topic, I guess, for further research and understanding about how the genes are perhaps passed on. Is it a recessive gene, for example, that only becomes active when both of those alleles are present. Yeah.
Yeah. So you can't tell by looking at the mushrooms or looking at where they're growing whether it's likely to cause paralysis or not. The question around whether a single patch is more likely to cause it again in future, we think probably yes, and that's because, like Kane said, I think we're pretty sold on the idea that it's likely to be genetics of that local population of mushrooms. Like, yes, the subs that are growing there are the same species as the subs that are growing down the road, but they can have genetic differences that can alter their chemotype, so the sort of amount of and the combinations of the chemicals that the mushroom is producing. And so it would make sense because, like Kane said, subs and Limcola species, they'll persist in that patch for years with often the same genotype over years until it sort of gets superseded. That makes us think, and certainly what we heard in the survey was that people picking from the same patch, if it's caused weakness before, there is a much higher chance that it will again, including in subsequent years. We do have a project on iNaturalist where you can upload locations where you've picked mushrooms that have caused woodlover's paralysis. Now, when we've mentioned this before, some people have been like, but then people will know where we pick our mushrooms. I say, well, my advice would be you probably shouldn't be picking there anyway, because there's a good chance it will continue to cause woodlover's paralysis. And like we say, that can actually get dangerous, even if it hasn't been before, because you never get, we don't know how much of this chemical is in these mushrooms. We don't know what affects that. Maybe the next batch is going to be heaps more potent at causing the weakness, and maybe that's what tips over the breathing issue. So two ways that's helpful to upload those patches onto the iNaturalist project for those paralysis. One is it allows access to a source of mushrooms that cause it for analysis to try and figure out what is actually causing this. And secondly, it helps warn other people that they probably shouldn't pick there either, because otherwise it's really, really hard to sort of, list where patches are that have caused it so that people can actually avoid them, which I think is probably more important than trying to protect a patch that, may not be safe to pick from anyway. Yeah.
A little
Thank
bit of
you.
speculation there, though, also on that point is, is it common? And I know kind of it's something that I've thought about and Simon and I have talked about is with people walking from patch to patch, spreading spores. Is there the possibility that the genes are being spread and therefore becoming more predominant within the population? And having something like the Woodlover Tracking Project potentially gives us that long-term kind of perspective to see whether it is turning up in more and more geographical locations.
Yeah, we've talked a bit about like what the potential function of this chemical that causes the paralysis might be to the mushroom, because evolution doesn't usually persist with things that aren't in some way useful. The hypothesis that psilocin and psilocybin are produced to sort of protect the mushroom against predation, especially from things like insects and slugs. Neuromuscular junction blocking type agents called anti-nicotinic neuromuscular junction blocking agents because of how they work at that gap between the nerve and the muscle. There are chemicals like that that are actually used as insecticides and pesticides because they do impact on things like insects and can kill them. So if the goal of the mushroom in producing some of these chemicals is to stop some of those types of creatures from eating the mushroom before it gets its chance to do its thing and grow to its full size and release its spores, which is what the happy little mushroom really wants to do. That's its one job in life, really. Then perhaps if this woodlover paralysis causing chemical is actually better at serving that purpose and protecting the mushroom, there is the possibility that spores spreading from patches that have caused woodlover's paralysis because they produce this chemical might give those spores and the mycelium they produce an advantage over other populations of subs that don't produce that chemical. And that may mean, and this is total speculation, absolutely not saying that this is what's happening, But that may mean that over time, it would be possible that the wood lover paralysis chemical producing populations might actually have an advantage in spread, especially since humans are sort of trampling through the bush and tracking spores all over the place, whereas before, spore transfer would have potentially been a little bit more limited.
Thank you, Simon. That's really, really fascinating. And I'll do my best to link the iNaturalist stuff in on Facebook so that everyone, people can take a look at that and maybe contribute to it. So really wonderful info about the genetic component of this. There's been a bit of sort of speculation that it could be related to contaminants. So is there any truth to the fact that it could be caused by something like herbicides, insects, or other colonizing microorganisms?
We feel no. And there's a number of factors there. Essentially, one of the questions we had in the survey, or a couple of the questions we had, were regarding substrate and habitat. And respondents reported wood lover paralysis being caused from populations across the board. So, and this includes native populations of subs that are out in the forest, out in environments where there's likely not to be herbicides being spread. So that for us kind of was pretty clear. And regarding the whole aspect of whether it was insects or other microorganisms, you would expect if it was insects and other microorganisms, there'd be the potential for wood lover paralysis occurring from other kinds of fungi that people are foraging. So given that this is just so specifically, but also other psilocybes as well for that matter, given that this is so specifically within that particular group of psilocybe section Seborrheicosa. So Seborrheicosa, Cyanescens, Azurescens, and even Psilocybe wurrawoa, they're all very, very closely related within a particular section. There have been reports from other species, and we're unsure of the relationship there. It could just be that those species still have part of that mechanism that's causing the compound, but we're not sure. And I guess this is kind of where full genome sequencing is going to help us better understand that aspect. But the fact that it's just within that particular little group of the genus Psilocybe and none of the other psilocybin-causing mushrooms kind of leads us to think that it's again it's a function of the mushroom itself.
Yeah, people wild forage a lot of edible mushrooms in the same habitat, same time of year, literally growing next to subs. Think Lactarius, think wood bluets, think different agaricus species. Quite a lot of forage mushrooms growing in the same locations at the same time of year as subs. None of them cause this. So it would be really weird if it was an environmental contaminant of any form.
Yeah, fascinating. And have you seen any correlation between, say, high nutrient density substrates as opposed to something with like a less nutrient dense, more sort of wild situation?
We kind of have, and I think this is a little bit of kind of we need to dive further into the environmental data that we got from the survey. So it's a little bit of a watch this space. But in general, a lot of foraging happens within the wood chip environments as well. So there's a little bit of a bias towards that. And I guess another aspect is that, as you've pointed out, people, the nutrients, it's anecdotally well known that the wood chip subs are a lot stronger than the native growing subs. So we would expect that subs that are being picked from wood chips there would probably be a correlation with And particularly if it is a tryptamine-based compound that comes from the psilocybin gene cluster, we would expect that correlation, in line with the increased amount of psilocybin and psilocin that is known to occur with mushrooms that grow from wood chips.
Thank you. And I know that the study focused primarily on Australian species, but someone in the chat has asked if it's equally present in other countries.
Yeah, don't know about equally present because we didn't get anywhere near as many responses from overseas as we did from Australia, which makes sense. We're advertising mainly in Australian groups. But certainly we had reports from Europe, North America, was it South
Canada.
Africa, Canada? Yeah. And there is a report from Japan as well, which was a less closely related species, which was interesting. And New Zealand. So it has been reported sort of more widely.
And I guess another aspect of that is just the recent research that's been looking into the origin of some of these species, Psilocybe cyanescens and Psilocybe azurescens, and highly likely probably Psilocybe eleni as well, which are known to cause wood lover paralysis, originated from Australia. So they are essentially Psilocybe tuberiginosa, and they have taken those, the genes that we assume the genes that cause the production of the compound are present within those populations. There is a lot less genetic flow, it seems, within those international species. So it's probably likely that it doesn't occur as often. Whereas in Australia, there's a lot more genetic movement between within subpopulations and populations.
Yeah, having said all that, like one of the reasons that they're not using Lignicola species in like Oregon as part of their sort of regulated non-medical therapeutic psilocybin program was around concerns for wood lover paralysis. So it's certainly not uncommonly known about over there. And most of the early reports, probably reflecting the demographic of the early users of shroomery, were from North America and Europe.
Thank you. And I think you touched on this one before, but it's been asked, are the mushrooms trying to kill us? Or what function does wood lover's paralysis serve?
No, the mushrooms are not trying to kill us. But I think as per, we were speculating earlier, there's a likelihood that these compounds do have an environmental basis, for a developing mushroom.
An environmental function, shall we say.
Yeah, an environmental function, yeah. That to deter predation on the mushroom while it's developing and growing, is a highly beneficial function. The intention may not be by the mushroom itself to kill something, but if it does stop, predation by a slug or something, and kill off the slug, I guess that is beneficial to the mushroom.
Absolutely. Thank you. So it's also been asked, someone's asked, could it be linked to eating some of the substrate or wood, even just fragments that are attached?
No, that's highly unlikely. And another part of the survey was questions around how people were preparing their mushrooms. So regardless of preparation, whether it was just eating the dried mushroom or fresh mushroom, preparation in a tea, or even using the lemon tech, people still experienced wood lover paralysis. So it would have nothing to do with eating of the fragments of the mushrooms, of the wood from the mushrooms.
Thank you, Kay. So we've had a question from one of the contributors over at PMANZ, and they were interested in finding out if there's a single causative agent, and if so, could a test kit be made that might indicate wood lover paralysis risk?
Well,
Um,
we don't know yet. Yeah, yeah, yeah.
yeah, no, we don't know yet. It could be a single causative agent, but then it's a matter of developing. I mean, there is the potential for a test, I think, if it is a single causative agent.
Yeah.
Um,
There's the quantitative sort of silo benchtop test kits now, aren't there?
yeah, there are. Yeah.
So if it was a similar tryptamine, like it might be plausible to be able to do that.
hmm But I mean, some of those test kits that are available are simply just looking for tryptamines, aren't they? So,
Mm.
in which case that's not very helpful.
Yep.
So, it would have to be a fairly targeted test, I think. There are some potentially interesting technologies out there, things like lamp tests that could potentially signal presence of the gene itself that does that does cause the compound. You know, but again, it's speculative, and there needs to be further research to find out, whether it is a single compound, and I guess then looking at other mechanisms of action as well for individuals.
Mm. Yeah, and at that point, like one has to question whether it's not just worth looking at alternative methods of sourcing product that's not going to cause it. If you want to be really safe, then looking at really complicated chemical kits to try and make sure that the ones you've harvested that way aren't.
Fantastic. You know, watch this space. We've got a question in the chat. So, does wood lover paralysis episodes affect the brain signals to the nerves, or is it a direct nerve ending paralysis?
Yeah, really interesting. My personal sense is that it is probably at the neuromuscular junction, so nerve ending, where nerve meets muscle. Reason I think that is because of the sort of reports that use of the muscle causes weakness, and then resting the muscle tends to regain some strength before use again. That's pretty similar to a syndrome called myasthenia gravis, where there are autoimmune sort of antibodies directed against some of the acetylcholine receptors in the neuromuscular junction. So one of the chemicals that causes the muscles to contract that's released from the nerve. You get antibodies that block that, and that causes a really similar use-dependent weakness that recovers with rest. Also, the distribution of the weakness is really hard to fit with a central mechanism, but really easy to fit with a peripheral, like nerve meets muscle sort of location mechanism, just because the way the brain's control of movement is organized is not sort of remotely similar to the way the weakness tends to present in terms of the pattern of where the weakness affects the body.
Thank you. We've got actually quite a lot of questions coming through in the chat at the moment. So someone's asked, so is it potentially more likely to come from pins rather than mature mushrooms if they're trying to prevent predation?
I feel that's probably is more likely from pins. I think the question there too is like, when are the genes regulating the production of psilocybin and other compounds? When are they being regulated during the formation, development, and growth of the mushrooms? It's, we kind of know that pins can be quite potent, so that perhaps maybe at a certain point the mushroom does stop producing psilocybin, but I think this is a little bit more of a kind of a research space. Yeah. It's hard to know at this point without further research into that upregulation of the genetic component around the whole psilocybin gene cluster, not just the compound that produces wood lover paralysis.
Yeah, and be super hard to know without knowing what the pathway to the production and what the chemical that is producing is to try and figure all that out. The other thing that's really interesting, and I think it touched on maybe one of the other questions in the chat, is that whatever the chemical that causes wood lovers paralysis is, it persists or seems a lot more resistant to inactivation by breakdown than does psilocin or psilocybin. So mushrooms that have been stored very poorly or dried very poorly to the point where they no longer have much psychoactive effect, i.e. the psilocybin is all oxidized and broken down, can still cause wood lovers paralysis. And there's a couple of interesting anecdotes that maybe even more likely to or cause it more significantly than they did when they were fresher or more properly preserved, which is interesting, thinking about whether there's some possibility that what causes wood lovers paralysis is actually like an oxidation product rather than something that is produced directly by the mushroom. So like psilocin, it oxidizes, it dimerizes, that's what causes this blue diquinoid sort of product that makes it stain. 4-hydroxytrimethyltryptamine, which is the N-methylated psilocin, so it's had another methyl group, which is a carbon and 3 hydrogens sort of tacked on to the end of it, where there's usually 2 methyl groups. It looks structurally very, very similar to bufotenidine that causes paralysis as well. The only difference is the position of a hydroxyl group on the ring on the other end of the molecule. A lot of the neuromuscular junction blocking agents are trimethylammonium compounds, so they have those 3 methyl groups on one end. And that sort of makes me speculate about whether that might be the culprit. That's certainly one of the things that hopefully someone will look into. But that compound, from what we know, probably does oxidize and probably does dimerize when it does that, because we've heard that solutions of that, like analytical solutions of that, do change color if they're not protected from oxidation, which would indicate probably a similar oxidative sort of pathway to what psilocin undergoes. A colleague of ours pointed out that what that product might look like looks very, very similar to one of the pharmaceutical neuromuscular junction blocking agents that are used to cause paralysis for surgery and in research. So older mushrooms might actually have more of that oxidation product in them than, you know, fresher ones, but that oxidation doesn't usually start while the mushroom is still growing unless it's been damaged by frost or rain or it's damaged, some other damage.
Oh, that's incredibly interesting. So while it's less common to find cultivated wood-loving mushrooms, there's been a number of people in the chat who have asked if it's showing up at all in cultivated mushrooms or is it just in wild or urban mushrooms?
There's a couple of ways people are cultivating them, and there's the most typical method is in, you know, outdoor beds. But I think that question is targeted towards the indoor cultivation methods. Indoor cultivation is really, really quite challenging. And yeah, it would be, I've certainly not heard any reports of people experiencing wood lover paralysis from indoor grows yet. Like, you know, like on a typical indoor cultivation substrate, so on grains or grains mixed with compost, for example, in the same sense that cubensis is grown. And that is something that would be a really nice study to be able to do in a controlled environment to then see whether a wood lover paralysis, you know, causing genetic individual grown in a typical cultivation environment does continue to produce the wood lover paralysis compound. My guess is yes, and quite possibly quite high because those mushrooms would be quite potent.
Yeah, certainly one thing that we'd, you know, like to see done as part of future work is, you know, controlled cultivation. So take a clone of a genotype that has caused wood lovers paralysis from the wild and then take it and grow it on different substrates in controlled indoor conditions and see whether that still produces the paralyzing compound even on different substrates in controlled conditions. And you could even try sort of messing with some of the other environmental factors that way in a controlled way, just to see whether there's, for one, that would sort of really prove that it is a genetic thing and not entirely a substrate thing, but it would also sort of demonstrate whether there's enough of a gene environment or a gene substrate or a gene habitat type interaction to explain the production of the paralyzing compound entirely, or whether that can just influence it. So that is to say, like, that mushroom that has caused it in the wild has the gene that makes it produce this mushroom, which may be an extra copy of like Psi-M that produces, say, that trimethylation of psilocin, or it might be something completely different. That's just what I reckon it might be at the moment. You take a clone of that, so you've got that genotype, those genes for producing that compound in there in those amounts and regulate it the way they are. So it can produce this paralyzing thing, and then you take it and you grow it on straight up pine, you grow it on straight up straw, and you grow it on straight up grain, and you grow it on straight up hardwood and just eucalypt and just this and just that, and see whether that influences how much of the paralyzing compound is produced on each of those different substrates, which is a lot easier to do once you know what chemical to look for when you're analyzing all of those things to compare them. That's the first step.
Wonderful. Thank you. So it seems to be like a lot of people are really interested in the research that you're doing, and Darklight in the chat has asked, what's the best way forward to get more funding for your research?
More funding.
And then he followed up with saying, or even some funding.
Yeah,
no, because we've done this off our own back. There's been no funding involved here.
Yeah. I reckon it'd be a real challenge, wouldn't it, to get funding for something like this, because I don't think there's likely to be a whole lot of commercial applicability. Like, commercial neuromuscular junction blockers already exist. There's a fair range of them. There's probably not a huge pharmaceutical appetite to come up with a brand spanking new one.
And university research these days does require some kind of output. There's not a lot of just let's just research it for the hell of it kind of research that happens anymore. It's really only if you're like in a particular position where you have the authority and the ability to push that funding.
Which luckily, like, thankfully and luckily, and just sort of a testament to the connections and the sharing nature and the curiosity of this whole community. We've managed to sort of form some partnerships with some people who are in positions to actually start doing some of that chemical work without any funding, just out of curiosity's sake and for the sake of harm reduction and interest, which is I think one of the really heartening things about this whole thing is the amount of people that have given up time either to complete the survey, to help us sort of think this through, to talk about it, now to move on to the next stages, just because it needs doing and because it's interesting.
And I think the community really appreciates the fact that you have put in this labor of love and followed this curiosity and been able to provide all this really important education. So really big thank you to you guys and everyone involved. So another question that's come up, are there any animal models of wood lover paralysis, or have any animals been observed to experience anything similar?
Super interesting. Not that I know of. I mean, geez, how you'd even spot that would be. Yeah.
This is something I'm really, really interested in. And I would love to speak to some ecologists or even just request that people in the community just keep an eye on slugs or other gastropods who are potentially feeding on the mushrooms, whether they any of these appear disabled or appear to be dying after having munched on said mushrooms. I feel this would be a really interesting thing to track.
Which would also be really easy if you had them in indoor cultivation in controlled environments. You know, you could actually do preferential predation sort of studies by introducing a predator and seeing which of two different genetic populations they preferred to eat and what the effects of them eating the, you know, paralyzing ones was versus the other ones, to see whether it does produce some sort of survival advantage. But I just want to go back on that point as well. CJ sort of asked when we were talking before about whether it's happened from indoor cultivated things. I think you're right, CJ, like very few people are cultivating Lignicola species indoors. So if they're in an outdoor patch, it's somewhat controlled, but not completely controlled. Be really interesting to hear from people if they start to experience this from their outdoor grows of these, from stem butts that they've transplanted or however they've gone about it. But I'm not sure. Kane, have you heard of anybody having that happen yet with someone with a patch they've actually kicked off?
No, not yet. Not yet, but I expect that we'll probably start hearing from people, given that these techs are becoming quite popular within the community. And that would be the community too that I would love to talk to about the slug issue. I've had a few conversations with people about how they've observed slugs close to the mushrooms who are not looking too good, but you know, it'd be nice to have something a little bit more concrete and less anecdotal about that.
It does sort of, it would, I suppose, make intuitive sense from a harm reduction standpoint. I'm certainly not advocating that anybody do anything illegal by growing these things because they are very illegal. But it probably from a harm reduction standpoint would make sense to say that if you were planning to do that, you use, you know, genes from a patch that you know, at least when you last picked it, did not cause paralysis.
I'm just having a bit of technical difficulty.
But yeah, no, I guess, and coming back to CJ's point, yeah, I mean these are really, really difficult mushrooms to grow in an indoor environment that have particular triggers that haven't been fully worked out yet. Like we all know that cold seems to be a trigger, but it seems like there's a little bit more to it in order to get these growing in a controlled environment. Some people have been quite lucky with their genetics, and there was a paper that came out from Oregon last year where a group had been cultivating subs indoors with a license in a controlled facility, and are doing chemical profiles of said mushrooms, subs, and other species as well.
Fantastic. Thank you very much. Oh, sorry. So another question's come through. So does psilocybin show up in a blood test, and would there be wood lover paralysis indicators in the blood?
Psilocin does show up in blood tests, yes, but you can't test for something if you don't know what it is. So you can't really look in blood for you know, a completely unknown chemical. So yeah, thus far it hasn't been useful. Like the coroners that have been done where it sounds like people have had wood lover paralysis before a death, they haven't found anything other than psilocin, but they also wouldn't, you know, you can't target what you don't know chemically, is there anyway?
Yeah, no, you need to know what you're looking for in order to be able to do these tests.
Thank you. And have the results of your study shown whether or not it's dose dependent?
How many questions did we ask in the survey, Kane? It was a lot. Man, it
Well,
was a lot.
yeah.
Yeah, but we forgot to ask what dose people were taking, which in hindsight is a pretty significant oversight. So all we can do is sort of speak from anecdote and like hearing lots of people talk this through. It sort of intuitively seems like it's probably going to be more common at higher doses, but that doesn't mean it doesn't happen with low doses. And like I said before, some people have reported having very, very poorly preserved specimens that have been essentially devoid of any psychoactive effect that have caused quite profound wood lover paralysis. And the other really interesting thing that I think would be good to think about when thinking about the possible mechanism and also in future research is that quite a few people anecdotally now have said that their experience of wood lover's paralysis has been when they've had repeat doses over a shorter time frame. Now, whether that's multiple doses across the single session in a 5-6 hour type span, or whether that's multiple doses across multiple sort of sequential days, enough people have said that that's when they've had their paralysis, that I think that warrants some consideration. There's a bunch of potentially very complicated mechanisms for why that might make it more likely to happen, but that would all be complete speculation in terms of how the body handles the chemical, and whether it's a metabolite or something like that that's accumulating, or whether it's having some sort of slightly different type of activity at the neuromuscular junction, either depleting some of the transmitters or changing the way it expresses some of the receptors, changes intracellular processing of electrolytes. There's heaps of different mechanisms. Moving a muscle feels really easy, but it's not simple. So I suppose it is possible that those repeated doses might also increase the chance, but it's all just sort of anecdote at that point. Certainly from low doses, from high doses. Kane and I were talking before. I don't know that I've heard about many people saying that they've had it from a straight up micro dose.
No.
Yeah.
No, we did have someone in the survey report that they were microdosing, but I think it was one of the individuals who didn't experience wood lover paralysis.
Oh,
There's nothing to say that, like you've pointed out, that people can potentially hold on to mushrooms for quite a while and over time, increase the dose for their microdoses. That could potentially trigger wood lover paralysis if you were taking like enough to keeping in mind, the breakdown of psilocin. So that you could potentially have a small amount of wood lover paralysis or more, depending on how much you've taken of your degraded material.
Interesting, looking into sort of storage and how that may impact it, and yeah, very interesting. So is there any indication that some people may be more predisposed to it than others?
Yeah, this is a super interesting question. So we did ask about some individual factors in the paper, but to, really at a fairly crude level, it was mainly around like, does this only happen when you're using other drugs or using other medicines or have a particular allergy or have a particular medical condition? We didn't find any sort of any indication at all that any of those things were particularly relevant, which isn't to say that some other medicines, medical conditions, etc., might not predispose you to an increased risk. It's just not something we picked up on in our survey. And obviously it can happen to people who aren't on any medications, don't have any significant medical conditions or any allergies. I think there are going to be things that increase people's risk because we know that some people are more susceptible to other neuromuscular junction paralyzing agents than other people for a whole range of reasons. If it is a metabolite that causes this, then potentially differences in individual metabolism might increase your risks. I have heard from one person who said that they had had this every time they'd had subs, and it didn't matter where the subs were from. They hadn't had it when they'd had cubes, and they'd had subs from a wide enough area that when one certainly wouldn't say that that was a single population. And it had happened every time, whereas other people who'd eaten the same ones usually didn't get it. So I suspect there are going to be some people who are more susceptible to it. The thing is, you'd probably never know. The vast majority of the people in our survey who had it had also taken subs without having it in the past. So it's certainly not only people who are susceptible, but I reckon, as with most things, there's going to be an individual component to it. We're just not going to know what it is without quite a lot more detailed work.
We've had quite a few anecdotal stories of people who've consumed the same, the same tea or the same batch, and one individual experiences wood level paralysis while the other doesn't. They just sit and essentially then help their friend through the process. So that is also kind of interesting that we are seeing these kind of examples, these cases.
So that feeds in really well to the next question, I think. So someone's asked, am I more likely to have it again if I've had it once?
Again, I think it's really hard for us to say. All we can say is that some people do seem to be more susceptible to it happening, but it can obviously just happen occasionally to people because that's what those particular mushrooms were going to do, and then the next ones won't. So I think it's really hard to know. It's good to know that if it happens to you regularly, it's probably a good sign that there's an aspect of you, providing you've taken them from different patches and stuff, and it hasn't all been from the same batch and the same patch, which means that you are at a higher risk, and then therefore probably at a higher risk of having some of the complications just by virtue of repetition and chance and luck. It may also mean that people who are more susceptible to having it are more likely to have it badly at higher doses. Or say, for example, you're someone who, let's say, for example, there is a very, very small amount of whatever this compound is in all subs, and it is just present at such a low level in the general subpopulation that it doesn't cause weakness for most people, right? And then occasionally different populations of the mushroom have more of that compound in them, and that takes it up to a level where it causes weakness in some people or most people who eat that batch from that patch. If you are the sort of person who for some reason is so sensitive to the compound, you get it from all subs, even those that have this present at a very, very low level, I would say it's pretty reasonable to assume if you're unlucky enough to eat subs from a batch that have a much higher level of that compound in them and cause weakness in other people who don't usually get it, there's a good chance it's going to be really severe for you. And again, really severe wood level paralysis comes with a significant risk of accident, injury from accident, injury from choking, not swallowing properly, and worst case scenario, injury from not breathing anymore, then death. So I think it, be something to try and be aware of yourself, that if this does happen a lot, even when it doesn't happen to other people, subs might be a much riskier game for you than they are for most.
Thank you. I think that's really, really important to keep in mind. We've also had a question. Is it true that taking an antihistamine can reduce the effects from what you found?
Oh, this is such a tricky one to tease out in a survey. So the antihistamine thing came from in my view, fairly incorrect theory on the mechanism that was posted on the Shroomery years ago. And that theory was basically that it was a histamine excess that caused this, under the assumption that it was some sort of allergic reaction to some sort of contaminant on the mushrooms, i.e. not the mushrooms themselves, but say a mold or something that had grown on them. You eat the mushroom that has the mold on it, you have an allergic reaction, your body in theory releases a whole bunch of histamine, and that somehow causes weakness. So an antihistamine should help. There's a few big problems with that. One is, it seems very unlikely that it is a contaminant to the mushroom. The other is that there are no other symptoms that present with wood lovers paralysis that would suggest a massive systemic histamine response. People aren't itchy, they don't have hives, they're not swollen up, don't have issues with their blood pressure that we know about. So all these other features that typically occur with systemic histamine reactions do not occur with wood lobe paralysis. So I don't think they're likely to help through that mechanism at all. The other thing is, yes, a lot of people anecdotally report that antihistamines help, but when we talk about antihistamines, it's important to recognize that that is a large class of different chemicals that have different structures, and almost none of them are pure antihistamines. Almost all of them act on some other receptors. But which other receptors is very, very different depending on which specific medication you're talking about. The newer non-sedating antihistamines act on a very different set of receptors and are actually much more specific to peripheral histamine receptors, i.e. not ones in the central nervous system, than are the older sedating antihistamines. The reason they are sedating is that they get into the central nervous system and act on histamine receptors in the central nervous system. That causes their sedation. They also act on a whole heap of other receptors in the brain, lots. A lot of the very old antihistamines are structurally quite similar to some of the very old antipsychotics. So they have effects on dopamine receptors and serotonin receptors, and also on some of the cholinergic receptors, but primarily the muscarinic cholinergic receptors, which are the cholinergic receptors that are in the central nervous system, not the ones that are present at the nerve muscle junction, which is where we think this is likely to act. So I've always thought that it was very, very unlikely, and I've always thought that the anecdote was about placebo effect and bias, expectation bias. I've read that this is likely to help, therefore I think it's likely to help, therefore it's more likely that I have a positive placebo response to taking it. Couple that with the fact that you're taking it in the midst of a syndrome that just by its natural course varies a lot, comes on after a random amount of time and goes away after a very variable and unpredictable amount of time. So how you line up cause and effect with anything that you've taken that you think has treated it is really, really hard to say, because for all you know, it was only going to last an hour anyway. And taking the tablet has done nothing, and maybe it was just going to stop anyway. For all you know, maybe because you knew it was there and it was so concerning that you actually took something to try and treat it, you might have changed other aspects of what you were doing that have actually stopped it from being as obvious, like rested. Maybe you decided you'd go and lie down. And we know then that you're likely to feel like you're less weak. So maybe it wasn't the medicine. Maybe it was the fact that you stopped walking around because you felt like you shouldn't be walking around because you realized what it was. So really, really, really hard to like disentangle all that stuff. And then really recently, because we kept hearing about it so much, I looked up the original one that was reported, which is diphenhydramine. Diphenhydramine is a very, very old antihistamine. It's not one that's sort of commonly used anymore because most people these days tend towards the non-sedating ones. Diphenhydramine in, I think it was like one study, one observational animal tissue-based study, did actually affected the effect of one of the neuromuscular junction blocking agents, like one of the ones they use in surgery. It did it in a fairly odd and unpredictable way. It wasn't as clear-cut as you take, or you put the diphenhydramine in the tissue and then the neuromuscular junction agent wears off quicker or doesn't work as well. It was sort of a weird response, but it did have some impact on the way this neuromuscular junction blocking agent worked. And that neuromuscular junction blocking agent works in a similar way to what I think, only think, don't know, but think the chemical that causes the paralysis works. So in that case, there might be some first principles support for the idea that diphenhydramine specifically, but not necessarily any other antihistamines, because it, again, is quite different structurally to most of them, might have some effect. But those are also animal tissue studies. That's not the same as human eat antihistamine, and then antihistamine do same thing as it does when I bathe a muscle in diphenhydramine solution and then try and bathe it in a solution of a neuromuscular junction blocking agent. There's a lot of other factors thrown into the mix between those sort of tissue studies versus real life effect. But It has always been anecdotally reported enough that it warrants further consideration and testing. All of that is going to be so much easier once we know what the molecule is that causes it and how it causes it. Then you can start to think about targeted treatments.
Oh, wonderful. And is there anything that's showing any promise outside of that for, maybe minimizing symptoms or for people that are having an episode?
I think probably really the best way to reduce your symptoms is just to relax. To not use the muscles that are being affected by the paralysis. We've had a number of people mention how, when they stop using their hands for a period of time, the paralysis essentially goes away. But interestingly, then when they start to use their hands again, that's when the paralysis kicks in again and affects the muscles. So, and this seems to be across the board with, hands, legs, eating. And I guess the breathing one is potentially probably interesting because that's kind of where things get really tricky because, we need to use those muscles and in order to breathe. So I guess probably that's the point at which you need assistance from other people. You should have let people know by that point. But yeah, if you only have a very, very low, mild case, just relax. Let people know around you know that you are experiencing will paralysis and just chill, deep breaths, take it easy.
Yeah. Yeah. Resting the muscles, resting all your muscles as much as possible, trying to stay as calm as possible because when you're not calm, you tend to breathe more quickly and more heavily, and that therefore you are using your muscles of respiration more. Staying warm, not trying to eat or drink because a lot of the time the muscles of the face are actually affected. So if you do eat or try to eat, there's a fairly significant chance you might choke. A lot of people actually report they can't even close their lips and operate their tongue well enough to drink liquids. And if you can't drink a liquid without a risk of choking, you definitely should not have food in your mouth. I know people that have had to actually physically scrape food out of their mouth with their hand because they couldn't chew it up any further. That's a really high risk thing. And even drinking, once it's that point, is probably too high risk unless somebody's so severely dehydrated that they will die if they don't, in which case they need to be in hospital anyway. Probably going without food and drink. Recovery position, because that's the easiest position in which to breathe. So left lateral, one knee over, so that you respire. If you're going to be in that position for a long time, you need to roll periodically. Otherwise, you get a pressure injury on one spot or the other. And never leave your friend alone if they are having this, because even if they look okay, it changes. It keeps variable. But like I say, I think if there's really any significant concerns, and particularly if there's any indication at all that someone's breathing is starting to get affected, you need to be on the lookout to start doing CPR as soon as it's necessary. And everyone who's using drugs or around anyone who's using drugs should know how to do basic life support, including CPR. And you should never hesitate to just call an ambulance if you have some feeling that something's just not right. 99% of the time things work out fine and everything goes fine and everyone goes home fine. But caution is preferable to dealing with what happens if something should have been done and didn't get done. None of that being medical advice, by the way. Nobody should be taking drugs. They're illegal and it's bad for you. But those who do should have the information to do so as safely as possible.
I appreciate that, Simon. That could be really life-saving advice. Really important to share that. Thank you. Another question's come up in the chat asking if you've noticed a specific cohort or demographic that's more susceptible than others to wood paralysis.
No, we didn't pick up on anything yet. I mean, we had a pretty mixed demographic that was pretty typically, I think, gender and or sex and sex and age bias, sort of in keeping with the places that we recruited for our survey from. But I don't think there was anything that popped up that made it easy to tell whether some people are more susceptible than others.
No, we just didn't have enough demographic information. Our questions around allergies, medications, and drug use were fairly general. And yeah, no, we didn't have enough information to identify any particular cohort. You would need a fairly large population study, I think, to be able to actually identify that. I mean, we were talking hundreds. I would imagine you probably need thousands
Yeah.
in order
Yeah.
to be able to identify if there is a cohort that is more susceptible.
Yeah. If anything, I suspect it would be easier to figure that out in reverse. Know what causes it, know what the mechanism is, and then link that to known factors that affect that aspect of, movement. Because a lot of this is, so neuromuscular junction function is pretty well studied because it is such a core component of surgery and the medications that are used in surgery. So once you know the chemical and you know the mechanism, it might be easier to figure out, whether there are certain, say, racial demographics that are more prone to experiencing, prolonged neuromuscular junction block with known surgical agents that you then think might be more susceptible to Willows paralysis and, that sort of stuff. Likewise around metabolism, like if it's a metabolite, knowing how the chemical is metabolized into the chemical that causes the paralysis, you can then potentially backtrack and work out whether some people whose metabolism, along that pathway is slower than for other people are more at risk. But I think really the first thing before being able to give anything more than very general symptom-based supportive sort of harm reduction advice is going to be to figure out what the actual mechanism is really.
Thank you. Thank you. So we're getting close to 9 p.m. So there's I do apologize if we don't get to all the questions. We've had so many questions come through. But I'd love to pass it over to Kane and Simon to see if there's anything that we haven't spoken about that you think could be really interesting or important to talk about to the community.
Hang on, I'm going to just have a look in the questions. I'm pretty sure we've covered all our kind of main points, the things that we kind of set out to cover with the survey.
I've got plenty of questions in the chat if you're happy to answer some questions that are coming through there.
I think one of the things for me is that, and it's fairly unrelated to Woodlover paralysis itself, but it's this whole sort of exercise of trying to work through this and figure this stuff out and try to form connections to get the next steps of work happening that are beyond our sort of skills and resourcing. Like, be curious and look into stuff. If something is really interesting to you and it doesn't seem like it's being looked into and it doesn't seem like there's, work happening in that space, figure out how you might be able to get the ball rolling. And if that means you doing something, awesome. If it means you finding some people who are interested who you can work with to get it done, then, awesome as well. And for me, this fairly long process, because it's been about 8 years since I first saw Woodlover's paralysis. Probably longer actually, probably closer to 10, and like 6 solid years of looking into it now. It's led to an awful lot of connections and knowledge growth and friendships and just cool stuff and opportunities. And so I think it's a really good example of the power of citizen science and the capacity to actually do this sort of thing, even if ordinarily you might look at something and be like, oh no, that's for someone else to try and figure out. That someone else might be you, or you might be able to convince somebody else that they should do it, even if you have to poke them a little bit.
Oh, it's really inspiring. You both pioneer in this area. Fantastic.
I know for me, a lot of this is around harm reduction, around trying to identify, this interesting syndrome that I don't think when we started looking at this, we realized just how problematic it could potentially be um, in the most severe cases. Um, but I'm fairly passionate about psychedelics and their potential uses in therapy and for wellness. And to have something that's kind of close to my heart in a way suddenly be implicated in something like this. It's like, well, I want to find out more about this. I want to dive into this and have a look at it. And that's been a big part of, you know, I guess it's striking up the conversation with Simon and going ahead and doing the work. Um, and for me, it's really just about communicating what we can about how to limit any potential harm that may come from these compounds. And these are compounds and mushrooms that um, have a really, really strong prohibition around them, and in some cases, really strong legal um, deterrence based around them. And we have the opportunity for the community to really kind of take this on and help educate each other and to reduce any potential harm around the compound itself. Like we don't want scare stories around this. We don't want there to be further prohibitions based on, a few people died from magic mushrooms kind of situation. Let's dive in. Let's understand this. I don't know, the powers that be, they're not interested in these kind of compounds in a lot of ways. So it's left to citizen science to really look into it and to further understand it. And it's been really, for Simon and I both, really rewarding to get this paper published and for people to finally kind of go, oh, this is a real thing. And how can we better understand it? Um, mm,
Yeah. And like in doing that, to be able to validate the experiences of people who have actually had this, who we still see get shot down on forum posts as just being, psychologically weak, or, you just can't handle your mushrooms, or they just needed to floor you to teach you some lesson. They can be really powerful therapeutic personal growth tools, but and it's 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 been such a fine line with a lot of concern about, does sort of bringing these sorts of potential harms to light increase the chances that there's going to be some sort of, increasing restrictions, or at least slow down of softening of restrictions. We'd all like to see at least decriminalisation, hopefully moving towards legalisation. Please check out the APS's decriminalisation position statement and discussion panel for more on that. But I personally think that, what really puts the brakes on those sorts of things is bad stuff happening, bad stuff getting either into the media or to the attention of the decision makers, and that bad stuff not being handled in a mature, open, sensible way by the people who are advocating for change. Denial of the potential harms or fictitious, exaggerated minimization of the potential harms does not look good when you are trying to persuade people on the other side of a position to you, because they already think these things are more potentially harmful than in reality they are. Pretending like there is no harm that may arise from their use does not convince them of that, and it never will. Going to them and saying, here's the evidence that those things you're worried about probably aren't as worrying as you think they are. Here's the evidence about some stuff maybe you didn't know about that we as a community have identified, we've looked into, and here's the solution package that we've come up with to that problem. We can actually address this stuff if there are and where there are risks and harms. we can actually maturely, scientifically, intellectually, academically, from a community-oriented place and collaboratively solve those and overcome them. And we've got the power and we've got the connection and we've got the community and the resources and the knowledge and the experience and the skills and the diversity to do that. And we've got the openness and the insight and the wisdom to tackle it openly and not to do it all sort of under the covers. This is what's led to so much blowback about some of the clinical research into MDMA overseas, is that it got to the end of it all, and then stuff that maybe had happened during the trials hadn't been handled during the trials as openly as it should be, and as openly as you'd expect that it should be in a clinical trial, because people were worried that if they started trying to address that midway through, the whole thing would get shut down. Well, what did that lead to? It led to it getting shut down after it was all done. And now you've got to basically go back to square one anyway, because they essentially invalidate all of the rest of the work, even if some of it was valid, because it looked like you were trying to cover something up. Whereas I think if we sort of can all work together to just address the risks and the potential harms openly, and go and say, okay, yeah, you're worried about this. Hey, we were a little bit worried about that too. Here's the response. Here's the harm reduction package. Here's the education. Here's what you might do if that arose in a situation where someone was using it. What's your next concern? Because we'll address that too.
Brilliantly said, Simon. There were some real wise words there. And so I think we've probably got time for one more question. So there's obviously the work you're doing has piqued a lot of interest and curiosity in the community. So I've got a question down here but do you guys have any plans to continue studying wood lover paralysis and trying to find out more about the mechanisms and possible compounds that cause it? Or do you think that after this paper and Q&A, you'll leave that up to other people to pick and study?
I certainly have an interest in continuing research in this. I am absolutely fascinated by the ecological and the genetic component of this. And it was a large part of working with Alistair McTaggart, for example, with all the psilocybe seborrheicosa work that he did over the past few years. And as a result of that, we have like 86 genomes of Psilocybe seborrheicosa that we can potentially start to look at to see if we can identify what gene may be responsible. But we also need a little bit more information about what the compound is in order to then be able to target what gene regions may be present. And I think there's also, wider phylogenetic information as well that may be interesting. Like, is wood lover paralysis the original defensive compound? Was psilocybin and psilocin maybe an afterthought by the organism? Or is it something new that's only just appeared? The mutation has proved interesting, and it's now spreading. There's heaps of questions. Yeah, I'm very excited to just keep looking and keep trying to understand.
Yeah, me too. I mean, as a case example of how sort of chemistry evolves in biological organisms, it's just fascinating. From the mechanism standpoint, from the chemical standpoint, I'm really interested, and also just as always from the harm reduction of the information spreading standpoint, I think this is really important. Like, I'm not that sure that identifying the mechanism and the genetics and all of that is going to largely change the harm reduction response, because I think ultimately, short of the potential opportunities to sort of cultivate from genetics that don't produce that thing, if you're picking in the wild, there's always going to be some chance of this happening. And I think when this happens, the response from a first responder is always more or less going to be the same supportive stuff and knowing when to call for help and knowing how to handle things until help gets there. But from a curiosity standpoint, definitely hoping to stay involved with, like I said before, collaborating with people who have skills and equipment and resources and knowledge that's way beyond anything I hold.
Thank you. I
And
personally really look forward to seeing where it goes in the future.
Thanks, Pip.
yeah, thanks.
Sorry. Kane, did you have something to add?
No, I'm happy to wrap it up there.
Yeah.
Um,
So yeah, big thank you to everyone that tuned in tonight. And a massive thank you to Dr. Simon Beck and to Kane Barlow, the mycologist and fungi educator, that you've been able to see and chat to tonight. Yeah, I hope that we get some more information as this starts to unravel, but really appreciate you and the team and everyone that's been involved in this study. And yeah, look forward to where it's going.
Thanks,
Thank you,
Pip.
everyone.
And thank you, Pip, for coming along. And again, thanks heaps for all of the work you're putting in with the APS. Our social media wouldn't look nearly as pretty or be anywhere near as active without your, well, running it. Put it that way. I was going to say help, but I had to rephrase. And also, Australian Psychedelic Society, entirely volunteer-run, donation-funded organisation. We did host the original survey. Please check out our website, psychedelicsociety.org.au. Please, if you're interested in volunteering, get in touch with us, info@psychedelicsociety.org.au. We're always looking for more volunteers and more people to get involved. If you are in a fortunate enough position to have a few bucks to spare, please consider making a donation via our website because that's what keeps us being able to do stuff. We've got chapters in Melbourne, Sydney, Adelaide, Brisbane, and some other smaller groups around the place as well, who put on sort of social gatherings and events, picnics, talks, workshops, walks. There's online integration circles. There's a women's group that runs once a month. There's a men's group that runs weekly. There are other special interest groups. So please check out our website, check out our events calendar, check out our Facebook and social media, and follow us and support us if you can, like you have by coming tonight. And for everyone who filled out the survey, huge appreciation. It was long. It was longer than it needed to be. So thank you for sticking it out.
Yeah, no, likewise. Thank you very much to all the participants in the survey. Thank you for, the time that you spent filling in the questions.
And Kane, do you want to maybe give a quick shout out for Garden States, because that is the other big news in the Australian psychedelic scene for this year and probably for
Yeah.
years to come.
So Entheogenesis Australis is holding a conference at the end of the year. So I think it's from the 28th of November to the 1st of December. It's going to be 4 days out in the Victorian bush, under the stars and amongst the trees. 4 days of talking around psychedelic science, ethnobotany, topics as diverse as, botany, mycology, philosophy, ethics. But coming back to community and our shared passion around psychedelic science and the plants and fungi we hold dear.
And that is gardenstates.org.
Yeah, thank you.
Awesome. Thanks everybody for listening. Thanks for all the excellent questions. Thanks for being so engaged. This video will be available on YouTube to watch later. So if anybody found it interesting and wants to share it around, please do. We've also got a whole bunch of other videos and stuff on our YouTube channel, so please check that out while you're there as well. If you have anything you would like to see more talks about, get in touch and we'll see if we can find the right people to give you the information you're looking for. On that note, have a good night, everyone.
Good one.